So developmental biology students, I've gone over this sheet with you in class about how to do immunostaining with monoclonal antibodies. And we've got our tissues ready on the microscope slides, but we need all these solutions as well. We've got some PBS buffer, which is phosphate buffered saline. And I've aliquoted some of that into a tube here, PBS. The saline is the salt solution, so it's got the right amount of salts so that the cells don't change their shape as I wash the liquid over them. I also have a tube with some PBS in it that we're going to make our blocking solution. The blocking solution needs normal goat serum. So this brown liquid, if you can see that in there, is the serum that contains antibodies that were floating around in any normal goat's blood. So those antibodies have no reason to bind to our tissue sections of axolotls here on these slides. So if those do bind, they will cover up any naturally sticky spots. They won't be binding to our protein of interest. They'll just be covering up any part that's sticky. I compared this in lecture to a lint roller, and our tissue is like the lint roller's sticky tape. And then we're washing all these little cat hairs all over it, and they're filling up all the sticky spots so that our antibody of interest, which is in this red liquid, this is supernatant from cells that were producing the monoclonal antibody that we want to use. So the supernatant means the solution that those cells were secreting the antibody into. They were monoclonal antibody producing cells that lived forever in culture. We studied those extensively. Those are hybridoma cells. And this is what they produced. And so we've got that pink liquid, which has our primary antibody in it. So first step that I need to do is let's mix the appropriate amounts of blocking solution. So I've got 250 microliters. Add this to the PBS. And now our blocking solution that I'm going to wash over these tissue sections will have enough antibodies on it that antibodies won't stick in random wrong places. Let me grab waste beaker when I'm done with the pipette tip. Use a smaller micropipetter to measure out the soap, the detergent. We're using Tween 20, and we need 15 microliters of this. Okay. Your protocol says you can use Triton or Tween 20. They are the same thing in terms of a soap that pokes small holes in the cell membrane of our fixed cells so that the antibody can get inside the cell and find the target protein that we want to study. Can I show you in the bottle here how thick that liquid is? It's like molasses. Okay? It's like thick dish soap. So when I go in to get 15 microliters of this, I have to let the micropipette tip, the micropipette depressor, this orange button, up so slowly, just barely releasing my finger. So now I have 15 microliters of this very sticky liquid. So watch what happens when I add our sticky solution to my tube. Can you see the liquid? I'm trying to stir it. Falling, falling, falling. It goes right to the bottom. And now I have to hold it depressed to make sure all the soap gets out. The soap is so sticky, it does not want to let go of my micropipette tip. So most of the soap is now jumbled at the bottom. If we come over here, we can use a vortex mixer to try to get that soap off the bottom and spread it throughout. So this spins the tube very, very fast just by my depressing it and now the soap has dissolved. And like, I only did this enough times that there's very few bubbles at the top. I don't want the bubbles, because bubbles on a microscope section will prevent the antibody from getting where we want it to get. So I'm going to take liquid from the bottom here, and hopefully that is well enough mixed with our PBS to keep the cells their normal shape, the blocking solution to make sure the cells aren't sticky so that antibodies don't bind where we don't want them to, and the soap so that there are holes in the cells so the antibodies can get in to find our protein. Now that we've made our blocking solution, the next thing to do is get our slides immersed in PBS. So to do that, we'll be using our Shandon Sequenza cover plates. So these come in a large box. You can see that we've got a lot of these left that you guys can use in your experiment. These cover plates come in boxes for specific use in these Shandon Sequenza staining racks. Okay? So there is room for 10 slides and cover plates down in this rack. What does the cover plate look like? I've got a box open here and a few of them taken out. This is what the cover plate looks like. It has a spring on the top. Notice how this part is bendable. It has a funnel at the top. Okay, so what is the top? When I put them in the rack, this is going to be the top. This is going to be the side that is on top of my microscope slide, and my microscope slide will be on the back side. So I'll take my microscope slide, and there are 6 little knobs, little notches that help guide this slide into the right place. Can you hear my fingernail clicking off of these? 1, 2, 3, 4, 5, 6. So my slide is going to brace against the bottom 2 and then be side to side. It can't move left or right away from, so those knobs are holding this in place. If you were to try to look at the space between where my tissue sections are on the slide and where the cover plate is, because it is completely flat and sheer on this side, only this side is a spring, there is basically no space between them. I have my microscope slide, my tissue sections, and then the plastic of the cover plate. They are all sandwiched perfectly together. Because water has cohesive and adhesive properties, when I put liquid up in the funnel region of the cover plate, it will evenly distribute throughout this tiny narrow space, completely submerging the entire slide. So, how do I want to get these two together? I've actually taken a couple cover plates and put one of them upside down on another. So now let me sit down here, grab some PBS, because we want to put them first in PBS. And I'm going to get a good bit of PBS here, more than we need. So that's probably about 1 ml of PBS, and we probably only need a few hundred microliters. Okay, so this is, double to 5 times as much PBS as I need. And what I'm going to do is I'm going to make sure I've got my end of my slide at the right place, and I'm going to use forceps to do this because my fingers are pretty fat. So where's the best forceps I can use? Got it there. So I've got a curved pair so that my fingers can be out of the way while I hold the slide. So I'm going to use the glass up against those notches and slowly lower it until the liquid starts to fill up the space in between my microscope slide and the cover plate. Can you see the liquid moving towards you as I slowly let it down? The liquid is now completely sealing all places in here. Because I let it down slowly, there was time for the liquid to push the air out of the way, and so this is completely covered with no air bubbles in it, meaning that the tissue sections that are sandwiched in there in the middle are completely engulfed, submerged in PBS. So what do I do with it? I get my Shandon Sequenza staining rack out, I press down on the slide and then grab it by the spring. The spring is going to apply pressure. Notice the PBS, that's extra. Remember I told you we used more than we needed to? We used so much more than we needed to that you can even see some of the PBS as I squeeze here is leaking back up into the funnel area. Now what I'm going to do is I'm going to place this in this direction so that I can lay my slide into the rack such that the rack will hold them squeezed together. So the spring is pressing right now to hold my microscope slide to my cover plate. But because there's pressure there, remember how capillary action works? Remember the properties, adhesive and cohesive properties of water? The liquid does not all leak out. It is all filling the space between those two flat plates. So if I do the same thing, you can watch it for my second slide. I'll get an excess of PBS. Just cover that thing, make sure it's nice and wet. Grab my second slide. Press the slide near down to the points. My liquid has started to flow, and I'm going to get my big fat fingers out of the way and slowly drop. And you can see the water marching its way up, displacing air until now this, I called it water, I meant liquid, right? The PBS is now filling that whole space in between, in the sandwich between my cover plate and my microscope slide. So I squeeze and drip. I'll have to flip it around because I'm holding it backwards. And place this one also in the rack. In terms of our procedure, we baked them, okay? We applied the cover plate with some PBS. Now we have to add 2 mils of fresh PBS. A disposable transfer pipette is graduated. Here's 1 mil. If you get liquid up into here, you've got about 2 mils. So all we're going to do is we're going to just press, squeeze the bulb, suck up as much liquid as I can, say, yeah, that looks like 2 mils, and squirt it in to fill up the funnel. Again, grab, sure, that's maybe 2 mils. The amount doesn't matter. All I am doing is making sure that the entire tissue sections, the entire set of sections that is on the microscope slide is going to be completely rinsed by this. So we're going to get rid of extra OCT, which we used in the cutting of the tissue sections to put them on the microscope slide. This PBS will be washing that off, and the tissue will be exposed to whatever other liquids we put in there. So the next thing we have to do, while this waits 5 minutes for that liquid to drip, for gravity to pull 2 mils of liquid through this tiny space in between the cover plate and the microscope slide. Well, let's use our time efficiently, and let's finish getting our primary antibody ready for putting as one of the next liquids here. So we have blocking solution. You watched me make this. It has soap and antibodies in it, non-specific goat serum antibodies. So what we need here is we need to take about 100 mils. So I'll use a 200 microliter micropipetter and set it for 100 microliters. I'll grab my blocking solution, and we're going to do, notice I'll put my tip way past any bubbles that are at the top because of the soap, so that I'm getting clean liquid with no bubbles. There's my blocking solution, and I'll just add it to a tube here. I've got a Eppendorf tube, a 1.7 mil Eppendorf tube. There's 100 microliters. Now, I'm going to use up everything that's in this tube. This is my antibody. Remember, this is the supernatant from the hybridoma cells. And I've got labeled the antibody here, and labeled by the company name and what product it is in terms of what it binds to. So I'm going to make sure that I can get this antibody. And look at all that pink liquid I've got. So I'm doing a 1-to-1 dilution, 100 microliters of antibody and 100 microliters of blocking solution. Antibodies are best kept on ice. It is bad for antibodies' stability to thaw them out so that they're unfrozen, then refreeze them, and then try to use them again. So even though I have a little bit of the supernatant left, I'm actually going to chuck this. Okay, so I do have my antibody now here. Notice that that liquid is also pink. You can tell there's a lot of that antibody solution in there. And we're going to use that to put on our slide that is ready for our antibody. And we'll do that as our next step. So we've applied fresh PBS into the funnels, and during that time we made sure our antibody was properly diluted so there'd be enough antibody to put on our slide. The next step will be to add blocking solution. Now remember, only PBS has gone through there, so these are still sticky to antibodies. We want to cover that up. So I'm going to use blocking solution, and we want the amount of liquid that can cover the entire internal space is probably about 100 microliters. 200 microliters is more than enough. So let's go ahead and use 300 microliters, and we will be absolutely sure that blocking solution gets into every nook and cranny by slowly being pulled by gravity down through our cover plate and the space between the cover plate and the microscope slide. So now it's hard to see how much liquid was in there because you know what? In that amount of time, the 300 microliters already flowed through. But it took 5 full minutes for the 2 mils to flow through. So what has happened here is the dynamics of the fluid flow are that I just put new liquid on top and there was PBS underneath. The new liquid pushes down the old liquid and replaces it. So we haven't mixed our blocking solution with PBS, we've replaced them because the liquid on top just pushes out the opening at the bottom, all the liquid that was stuck by capillary action in between the cover plate and the slide. So that 300 microliters is now being held by capillary action in our space and covering our slides. So now we need to wait an hour or more so that we have complete penetration of our soap, making holes in every single cell, of the non-specific antibodies in the serum binding everywhere that is sticky on those cells, so that when the next step, when we come back after an hour and put our antibody on just this slide and not on this one because we have a negative control, then the antibody, if it binds, we'll know it's binding not just because the tissue is sticky, but it's binding because it found its actual antibody target. So we are done with over an hour of blocking, meaning that the blocking solution has been slowly moving down through. Now I'm going to remove one of these just so you can see. It is still wet down here, and if we look at the back side, there is still liquid completely filling this area, but the funnel is now empty. So our tissue sections are facing the cover plate, and the space between the cover plate is still full of blocking solution. So I can see that none of those tissue sections have dried out, despite us leaving them at room temperature for an hour. Now we're going to put the antibody on one of our slides, the slide that is our actual experiment. So I'm going to grab all 200 microliters of this mixture of antibody in blocking solution. And I'm going to put that into the funnel on our antibody slide. Can you see the pink liquid, right? That there's a line of pink liquid across the bottom there. Right there. See the pink? It's starting to float its way down in. What do I need to do in the other one? Well, we want it to be as exact a negative control as possible. So I'm going to take blocking solution, which was the 50-50 solution of what we just put in, and I'm going to take a few hundred microliters of blocking solution and put that in our negative control. So no antibody has been added to our first one, our negative control, and antibody has been added to our second one. Now, because we want this to give this as long a time as possible for the antibody to find all the little holes in the cells and find our molecule that we're interested in and have the antibody actually bind to it, we're going to leave this overnight at 4 degrees Celsius, so in the refrigerator. And it also, by keeping it cold, the liquid, there will be less evaporation, so we don't have to worry about these drying out. I've got a lid on it and everything. We'll just put these in a refrigerator where they will stay until tomorrow, when we can get them out and see whether any of the sections have dried out, which we wouldn't expect, and then we can put our secondary antibody on. So next time we do videos, I'll show you how to put the secondary antibody on these.